Today's screencast is a little bit of a history lesson about how DNA was determined to be the macromolecule that stored and transmitted genetic information. So in order to figure out what was going on with genetics, they had to figure out what the gene is made of. They figured out what molecule carried genetic information, then they could do a little bit more research about how inheritance is controlled. The beginning of that discovery started in 1928. Scientist Frederick Griffith was trying to figure out how certain types of bacteria produce pneumonia in the organisms they infect. He took a look at two different strains of the same bacterial species. A lot of people say strands of a bacterial species, but it's two different strains of the bacterial species. These strains grew as they were cultured, but only one of them caused pneumonia. The S strain would make people sick, cause pneumonia. The R strain, people would stay regular. Now, S didn't stand for sick, it actually stood for smooth colonies. The harmless bacteria, the R strain, produced colonies with rough edges. So S and R stood for smooth and rough. I just use sick to help you remember which one's which. The S strain caused pneumonia, made people sick. He experimented on mice. So when he injected mice with the S bacteria, the disease-causing bacteria, the mice developed pneumonia and died. When he injected mice with the harmless bacteria, the mice stayed healthy. No surprise with these results. Then he started getting a little tricky with things. He took a culture of the disease-causing bacteria, the S strain, and heated them to outside their normal temperature range to kill the bacterial cells. So he injected those heat-killed bacteria into the mice. The mice survived. So the heat-killed bacteria did not cause pneumonia, even though it was the disease-causing bacteria. Did not cause pneumonia when it was heat-killed. In the next experiment, he took that heat-killed disease-causing bacteria and injected it together with live harmless R bacteria. Now the mice developed pneumonia and many died. So what happened was, we have the heat-killed S strain that would make mice sick, they were dead. The live R strain, for some reason the mouse dies of pneumonia. So something in that heat-killed S strain turned the living bacteria into disease-causing bacteria. The question was, what did that? If the S strain cells were dead and the R strain, the harmless bacteria, was alive, what happened? Griffith's conclusion that that there was some chemical factor that could change harmless bacteria into disease-causing bacteria, and that factor was transferred from the dead cells to the live cells. He called this process transformation because one type of bacteria had been changed permanently into another. He further concluded that that transforming factor had to be a gene because it was passed on to offspring of the transformed bacteria. Avery took the baton. Oswald Avery, another biologist, took the baton at this point. Wanted to determine which molecule specifically did the transforming. So he took a look at the different molecules that you guys learned about in Chapter 2 and treated that mixture of molecules with enzymes that destroyed proteins, lipids, carbohydrates, and nucleic acids. With each of these, even if all of these different macromolecules were destroyed, transformation still occurred. So we ruled these out. Not we, Avery. Ruled these out because even when they were destroyed, transformation still happened. When they repeated the experiments using enzymes that would break down DNA, transformation didn't occur. So his conclusion is that Griffith said, okay, something's going on, we call this process transformation. Some factor is turning living bacteria that don't cause disease into bacteria that do. Avery looked specifically at the different macromolecules and said, okay, well, the transforming factor specifically is DNA. Hershey and Chase did an experiment to further confirm that DNA was the transforming factor. They studied viruses. Viruses are non-living particles that can infect living cells. Specifically, they looked at bacteriophages. This guy looks like a little virus. If you used to watch Jimmy Neutron, there was an episode about viruses that looked like this. These are bacteriophages, which means bacteria eater. They infect bacteria. Does not infect humans, unfortunately. It made a better story because these are cooler looking than the viruses that infect humans. But, yeah. Jimmy Neutron took a little liberty there with that. When a bacteriophage enters a bacterium, it attaches to the surface of the bacterial cell and injects its genetic information into it. So I'm going to try my hand at a drawing here. Here's a bacteriophage. Here's the genetic information inside the bacteriophage. In order to infect the bacterial cell, that genetic information, now we know it's the DNA or the RNA that makes up the genetic material of the bacteriophage. But again, we were trying back in the day, trying to figure out, okay, was it the protein outside, the protein code of the virus, or the genetic material inside? The viral genes that are injected into the bacteriophage are replicated using the bacterial cells' replication DNA protein making machinery. Lots of different bacteriophages are produced. The cell splits open, it bursts out hundreds of new viruses. Like I said, I made reference to before, Hershey and Chase studied a bacteriophage that had a DNA core and a protein coat. They were trying to figure out whether it was the protein coat or the DNA core that was the transforming factor. Their results would either support or disprove Avery's finding that DNA was the transforming factor. We have two different bacteriophages here. On the left-hand side, we have radioactive phosphorus in the DNA inside as the genetic material. On the right, we have radioactive sulfur in the protein coat. The radioactivity is represented by the little orange dots. You see it inside on the genetic material here, the DNA. You see it outside on the protein coat all around here. So proteins contain almost no phosphorus and DNA contains no sulfur. These different elements could be used, if they were made radioactive, used as markers to determine which molecules actually entered the bacteria and had the bacterial cell produce more virus particles. If they found radioactivity from sulfur in the bacteria, it meant that the protein coat had been injected into the bacteria. If they found phosphorus, then the DNA core had been injected. So they ran two trials of this using the two different viruses, the two different parts being radioactive. A couple of different results happened. So here we have the bacteriophage with the radioactive DNA. After the infection, radioactivity was found inside the bacterium. On the right, we have sulfur in the protein coat, and the bacteria was not radioactive. The radioactivity in the bacteria was found from phosphorus, the marker found in DNA. So they concluded, along with Avery, that the genetic material of the bacteriophage was DNA, not protein. DNA has to be able to store, copy, and transmit genetic information in a cell. The analogy that we're using here is how you would share a book. Genes control patterns of development. They're an instruction manual to develop all living things. They have to be written into the DNA, these instructions. Before a cell divides, as we learned last semester, it has to make a complete copy of every one of its genes, similar to the way a book is copied. It was really confusing how DNA could copy itself. Once the structure of the DNA molecule is discovered, a copying mechanism was put forward. We'll get to that in future sections. When a cell divides, each daughter cell has to receive a copy of that genetic information. Sorting to make sure that there's a full set of chromosomes or a full complement of DNA is super important in meiosis when we're forming gametes. The loss of any DNA during meiosis might mean loss of genetic information in the next generation.